Most flu cases, around 70% of reported cases, are influenza A. However, the influenza B virus is the cause of the next most common type of flu. Influenza can affect humans and animals, but influenza type B is one type of the illness that causes symptoms in humans only. Also, if you're wondering which is worse, influenza A or B, type A is responsible for more severe illnesses. The symptoms of influenza B are similar to those of influenza A, and both types of flu can range from mild to severe. While influenza B is less common, it can cause more serious symptoms among young children. Symptoms of influenza B can occur anywhere from 1 to 4 days after exposure to the virus. Anybody can develop influenza B, but certain factors can also increase your risk of illness. For example, having certain health conditions can compromise your immune system and make it difficult for your body to fight off the flu quickly. Vanderbilt University Medical Center (VUMC) researchers have identified potent monoclonal antibodies against influenza B, showcasing a promising avenue for treatment and preventative strategies in combating the virus. Small molecule drugs that block neuraminidase, a major surface glycoprotein of the influenza virus, can help treat early infection, but they provide limited benefit when the infection is more severe, and they are generally less effective in treating influenza B infections. Thus, another way to combat this virus is needed. Reporting in the journal Immunity, the VUMC researchers describe how, from the bone marrow of an individual previously vaccinated against influenza, they isolated two groups of monoclonal antibodies that bound to distinct parts of the neuraminidase glycoprotein on the surface of influenza B. One of the antibodies, flu B400, broadly inhibited virus replication in laboratory cultures of human respiratory epithelial cells. It also protected against influenza B in animal models when given by injection or through the nostrils. The researchers suggested that intranasal antibody administration may be more effective and have fewer systemic side effects than more typical routes, intravenous infusion or intramuscular injection, in part because intranasal antibodies may trap the virus in the nasal mucus, thereby preventing infection of the underlying epithelial surface. These findings support the development of flu B400 for the prevention and treatment of influenza B and will help guide efforts to develop a universal influenza vaccine. Antibodies increasingly have become an interesting medical tool to prevent or treat viral infections. Researchers set out to find antibodies for the type B influenza virus, which continues to be a medical problem, and we were happy to find such especially powerful molecules. Thank you for watching. If you've enjoyed our videos, subscribe to our channel, for there's more to come.